Dogs seems to be much more reliable for that because we share similar pathway on the drivers of aging. The same biological pathways are used by dogs and humans. There has been very recently, in the last 12 months, several papers in Nature and in key peer-reviewed journal that demonstrate that the dogs are one of the best.
Hello, you're watching Proactive. I'm joined by Genflow Biosciences CEO, Dr. Eric Lyra. Eric, very good to speak with you. You've reached a safety milestone with your dog trials with no adverse effects reported so far. How important is this safety confirmation for progressing GF1004?
In terms of safety, super important. Why? This is a product that we expect will extend the lifespan of dogs. So it's not treating a disease. So it was absolutely crucial to have a perfect safety profile. Absolutely no side effect, absolutely no adverse event, very friendly for the administration of the dogs, easy to administer for the vet, and we demonstrated that. Any other response would have been a no-go for us because then we didn't have a commercial product. So this safety and feasibility is extremely important. Now we're not done. We still have to demonstrate efficacy, but we're also very convenient. We're very confident that we will get some efficacy data based on the previous mice study we conducted. What we're doing with the dog is to replicate what we've done with the mice. It's easier to see the safety with the dog than with the mice. The behavior of the dog is, we are more familiar with that.
Eric, what key results are you expecting from the 5-month follow-up, and how will you measure success?
So, first, we had to demonstrate safety, done. We had to demonstrate feasibility for the vet, done. Now, efficacy. Efficacy, we have multiple endpoints. One of the endpoints is to extend the biological age of the dogs. There's multiple ways to measure not your chronological age, but your biological age, your true age. And the best method is through methylation of certain sites of your DNA. Okay, CPG sites, or the technique, the people who love the techniques. The best test has been developed by Steve Horvath at UCLA, and we will use that test. So we measure the biological age of the dogs, of each dog at the beginning of the study. In 6 months, we will measure the biological age of the dogs at the end of the study. The dogs that are the control group are supposed to age normally, and they will have 6 months. They will be 6 months older. We hope our dogs to be either younger or didn't age that fast. Additionally, we have other endpoints that we measure. We measure sarcopenia. Sarcopenia is very important. It's the muscle of the dogs. Very important that your dog doesn't lose his muscle because then he's not moving. And you've seen the old dogs, they love to jump, they love to walk, but when they age, it's difficult. They just, it's sad. And it's very important for the dog to keep on to be active, to be able to walk. And that's one endpoint I will measure. Not only will measure this for the dogs, but we'll measure specifically the mitochondria in the muscle of the dog. So we'll take a microscopic, really small biopsy of the muscle of the dogs, and we analyze the mitochondria. We don't do that to hurt the dogs. That's the only things aggressive we will do, and it's a really small puncture for all the dog lovers. But it's very important to see if we ameliorate the mitochondrial functions. So we'll see after with an electronic microscopic, and we'll measure the improvement of the mitochondria. This has also implication in human. In addition to the biological age, to the mitochondrial function and the muscle mass, we have several endpoints like the quality of the coat, The hair tend to get really bad on the mice. We had fantastic results. Our mice were looking really good when they were treated. So we hope to have the same thing. Plus several endpoints, the classical marker of aging, the telomere, the senescent cells. Okay, but those are the key endpoints we want to achieve. We have multiple charts at a success.
Eric, what would the financial impact be of a positive outcome to the next phase?
Good question again. Very important. Um, we are in a situation where fundraising is difficult. The terms are not very good. Money is quite expensive for biotech these days. Therefore, we are favored by our model, relying a lot on grants, but you need to match the grants with some money. Um, Fundraising right now is not our key priority, and we would favor more non-diluting source of financing. Licensing out a drug like the drug we developed for the dogs with animal health companies is the perfect example. A positive dog trial will allow us to have a deal with a deal. When I say deal, it could be a licensing out, it could be a pure sales, but will allow us to bring non-diluting money in the company and to finance short-term the company, not using loans, not using convertible, not using even plain equity at very low, with a discount on the share price. That's what we observe these days. So to answer your question, yes, the impact will be extremely important in terms of finance for the company.
Eric, how might the data from dogs translate into human applications, and when could investors expect progress on that front?
That's a very good point you raised here. Um, it's it's important for the dog lovers to have a product that could at least extend the lifespan, how well their dogs are living when they get older. But it's also something we can extrapolate for human. The extrapolation from data from mice to human is kind of not strongly reliable. So when we say a product has an efficacy on mice by extending, the lifespan or increasing the telomere length, will be reproducible in human? Question mark. Dogs seems to be much more reliable for that because we share similar pathway on the drivers of aging. The same biological pathways are used by dogs and humans. There has been very recently, in the last 12 months, several papers in Nature and in key peer-reviewed journal that demonstrate that the dogs are one of the best animal to predict the efficacy of a product in humans. So not only the positive result will be fantastic for the dog lovers, for the dogs, but also it will give us a lot of advantages in terms of development in human.
Eric, I hope you'll continue to keep us updated with your progress on that trial. Thank you very much for speaking with us today.
You're very welcome.
That's Dr. Eric Lyra. He's the CEO of Genflow Biosciences.